Today, we're diving into a medical mystery. It's about one of our most powerful, most advanced cancer treatments, a therapy that, honestly, works miracles. But what happens when that miracle cure, in a 1 in a million case, turns around and becomes the cause of a brand new disease?
I mean, just think about that for a second. You're fighting a deadly cancer, right? You get this revolutionary, life-saving therapy, and it works. The cancer is gone. You've won. But then, just a few months later, you get diagnosed with a new cancer. And the truly terrifying part, it looks like it was created by the very cure that just saved your life. This isn't some sci-fi plot. This is a real-life medical mystery that we're going to unpack.
Okay, so to get what went wrong, we first have to get what's supposed to go right. And that means understanding the treatment itself, CAR T-cell therapy. And let me tell you, this stuff is a total game changer. Here's the deal. Doctors take your own immune cells, your T cells, out of your body. They take them to the lab and genetically re-engineer them. They basically install a new piece of software, this chimeric antigen receptor, or CAR for short. You can think of it like giving your T cells a super-powered, cancer-seeking GPS. This CAR trains them to do one thing and one thing only: hunt down and destroy your specific cancer cells. It is the definition of personalized medicine. All right, so let's get into the story. The patient in this case was a 51-year-old man. He was battling multiple myeloma, which is a type of blood cancer. He gets this cutting-edge CAR T-cell therapy, and at first, everything goes perfectly. The results are amazing. But that incredible victory, well, it was tragically short-lived.
So here's how it all went down. On day 0, the patient gets the infusion of his new supercharged T cells. Everything is looking good. But then, fast forward about 6 months. On day 190, scans pick up something new, these highly active lesions. Just 6 days later, he starts getting these troubling skin lesions on his face. And by day 200, the diagnosis comes in, and it is an absolute shock. Now, this is the really critical part. His original cancer hadn't come back. This wasn't a relapse. No, he had developed an entirely new disease, a rare and aggressive T-cell lymphoma. A brand new cancer had literally taken root right after the old one was wiped out. And you can just imagine the question racing through his doctors' minds. What on earth just happened here? So, this kicked off an intense medical investigation. I mean, the doctors had to become detectives, but their crime scene was at the cellular level. They needed to figure out, and fast, what this new cancer was, where it came from, and most importantly, how in the world to fight it. And when the analysis came back, it was a terrifying discovery. The cancer cells, they were the exact same CAR T cells that had been engineered to save his life. The cure had literally become the cancer. The official diagnosis was a CAR-positive double-negative T-cell lymphoma. And true to form for this type, it was aggressive, which explained why it was showing up as those lesions on his skin. So with this totally shocking diagnosis, the race was on. You can't just throw standard chemo at something like this. It's a completely unique cancer. The research team had to go on a hunt for a specific weakness, some kind of Achilles heel in these rogue CAR T cells that they could actually exploit. And their investigation was incredibly methodical. Step 1, they used this powerful technique called single-cell RNA sequencing. Think of it as getting a super detailed genetic blueprint of what makes these cancer cells tick. And this deep dive turned up a massive clue. The cancer cells were just covered in a protein called CCR4, way more than normal. Step 2, they had to find a weapon. So they tested a whole library of drugs against the tumor cells to see if anything would stick. Now, this discovery of CCR4, it was a real double-edged sword. On the one hand, it was a breakthrough. It explained why the cancer was attacking his skin. See, CCR4 basically acts like a GPS that tells cells to go to the skin. So great, they had a potential target. But on the other hand, finding high levels of CCR4 in this kind of lymphoma, well, that's usually a sign of a really, really poor prognosis. The clock was ticking, and it was ticking fast. Let's just stop for a second on this number, 166. That's how many different FDA-approved drugs the researchers screened against the patient's cancer cells. This wasn't just guesswork. This was a systematic, brute force search for a needle in a haystack. They were looking for any compound that could shut these rogue cells down. It just shows you the incredible scale of the effort to find an answer when there wasn't one in any textbook. Okay, so now they had these critical clues, right? They had the CCR4 target, and they had the results from that massive drug screen. Armed with that information, the medical team could finally build a battle plan. They could move away from a one-size-fits-all approach and design a treatment custom-tailored for this one patient's very unique cancer.
And the strategy they came up with was just brilliant. So, problem number 1, the cancer cells I covered in that CCR4 protein. The solution? A drug called mogamulizumab, which is an antibody that specifically hunts down and targets CCR4. Then, problem number 2, what else works? Well, the drug screen showed the tumor was sensitive to a class of drugs called anthracyclines, so they added those to the regimen for a powerful 1-2 punch.
And the result of all this incredible personalized detective work? It worked. It absolutely worked. The patient went into complete remission. This custom-built targeted therapy wiped out the rogue CAR T cells, and the best news of all is that as of the last follow-up, he is still cancer-free. Just a truly remarkable outcome.
But wait, this story isn't quite over. I mean, saving the patient was priority number 1, obviously. But the huge scientific mystery was still hanging in the air. Why did this happen in the first place? What was the genetic trigger that flipped the switch and turned these healing cells into cancerous ones?
So the researchers went back to the genetic data, and they found it. The smoking gun. It turns out the root cause was a 1 in a million genetic accident that happened during the manufacturing process. This mistake was made back in the lab when the T cells were first being engineered, before they were ever put back into the patient's body. So let's break this down, because this is fascinating. The cancer-fighting CAR gene, it has to be delivered into the T cells somehow. They do this using a harmless, disabled virus called a vector. You can think of it like a tiny biological delivery truck. Well, in this case, the delivery truck went to the completely wrong address. The vector inserted the CAR gene right smack in the middle of another really important gene called TIA1. Now, TIA1 is what's known as a tumor suppressor. Its whole job is to pump the brakes if a cell starts to grow out of control. And by parking itself right in the middle of TIA1, the CAR vector basically broke the brakes, disabling one of the cell's most important safety features. But just disabling that one safety gene, that wasn't the whole story. Breaking TIA1 seems to have made the cell unstable, kind of opening the door for more mistakes to happen. And that's exactly what happened. The cell then picked up new mutations in 2 other genes, TET2 and EZH2, and both of these are well-known bad actors in blood cancers. So you see, it was this cascade of errors, like one domino knocking over the next, that finally pushed this engineered cell over the edge and made it fully malignant. So, what's the big takeaway here? What does this incredibly rare but incredibly important case mean for the future of cancer treatment? Well, it's a powerful lesson, right? It shows both the amazing promise and the potential hidden dangers of our most advanced therapies. And the good news is, what we've learned from this one case is already helping scientists figure out how to build even safer cures for tomorrow. When you boil it all down, there are a few really crucial takeaways. First, these secondary cancers from CAR T-cell therapy, while extremely rare, are a real thing. It's a risk, even if it's a small one. Second, this case proves that when these crazy rare events do happen, doing a deep dive into the genetics isn't just an option. It's the only way to find a real solution. Third, it's an amazing showcase for personalized medicine. It can not only cure the first disease, but it can also help us solve the complex new problems our treatments can sometimes create. And finally, it just hammers home how important it is to keep monitoring patients long after their therapy is done, just to catch any potential issues early. You know, this whole case really leaves us with a big, profound question. We're creating these therapies that are more powerful and more personalized than anything we've ever had before. But as we keep pushing the boundaries of what's possible in medicine, we're also uncovering new risks we didn't know were there. So the challenge for the future isn't just about inventing new cures. It's about understanding them so deeply that we can perfect them, making them not just powerful, but also predictable and ultimately safe. How we answer that question, well, that's going to define the next generation of medicine.